will illuminate us more on the topic. So, without delay, let's proceed our event. Uh, I will ask Dr. Shapna Mukherjee, the State President of Microbiology Society of India and also Head of the Department, Dinobandhu Andrews College, to inaugurate the program. Madam, your speaker is off.
Can you hear me now?
Yes, madam. Yes, it's fine.
Thank you, Vipasha ma'am.
Please inaugurate, madam.
A very good morning to all.
Welcome, madam.
Respected Principal Dr. Shubhashis Datta sir, Honorable Chief Guests and Speakers, Mr. Sudhir Sahu of Google San Francisco and Dr. Abrojyoti Ghosh of Bose Institute, Kolkata, convener of today's webinar, Professor Shubir Dasgupta sir, faculty members, distinguished guests, and my dear students. A very warm welcome to all of you to this webinar on science, technology, and innovation in shaping education, jointly conducted by Maulana Azad's College and Microbiology Society India, or MSI as we call it in short. The society, many of you may know, started its journey 25 years back under the leadership of Professor Arvind Deshmukh of BSM University of Maharashtra, is now the largest microbiology society in India with about 5,000 members from colleges, universities, research institutes, industries, and medical field of India and abroad. Today, on this bright sunny morning, we have gathered to celebrate the National Science Day to show our dignity and respect to the famous Indian physicist, Dr. C.V. Raman, to mark his discovery of the Raman effect, for which we know, as Bipasha has already mentioned, he was awarded the Nobel Prize in Physics in 1930. And it is indeed a matter of great pride for us because he worked this entire tenure of about 26 years, starting from 1907 to 1933 at the Indian Association for the Cultivation of Science, which is situated at Jadavpur of Kolkata, doing research on many topics of physics, of which the Raman effect became his greatest success, which eventually wins him the Nobel Prize. In India, every year, the National Science Day focuses on a particular theme. And this year, as Bipasha has already mentioned, that it has been Future of Science, Technology and Innovation, Impacts on Education, Skills and Work. We may wonder why such celebration every year? What is the purpose? Well, then I may quote from Government of India Ministry of Science and Technology website, which says, quote unquote, to communicate science, technology to masses, stimulate scientific and technological temper, and coordinate and orchestrate such efforts throughout the country. I repeat, to communicate science and technology to masses, stimulate scientific and technological temper, and coordinate and orchestrate such efforts throughout the country. That is to popularize science and promote scientific thinking among all. As Bipasha has already mentioned that the whole world has now started readjusting or recalibrating to a post-COVID system, it is evident that science, technology, and innovation is the key to provide indispensable tools to help overcome the current situation and also prepare ourselves against any such future pandemics. I think today's discussions will throw some light on the existing and innovative measures to effectively respond to the challenges and opportunities offered by science and technology and innovations. I congratulate organizers of Maulana Azad College again for taking such a wonderful initiative to celebrate this day in collaboration with MSI in order to public understanding and trust in science. Looking forward to listen to our speakers with this hope. And again, I welcome you all. And with this, I end here. Thank you all.
Pasha,
Yes, sir. Now I request Professor Shubir Chandra Dasgupta, IQAC Coordinator and Head of Zoology Department, Maulana Azad College, to say something about the event and introduce today's speakers.
very good morning to all of you. Uh, on behalf of Principal Maulana Azad College and myself, Dr. Shubir Chandra Dasgupta, is a coordinator of DBT Star College scheme and also the head department of zoology. I welcome all of you. So before I say something, so I just remembering this importance of this National Science Day. So respected Principal Maulana Azad College, who will join shortly, just inform me, Dr. Shapna Mukherjee, State President of Microbiology Society of India, and also Head of the Department, Dinabandhu Enrich College. Our two honorable speakers, Mr. Sudhir Shahu, the Senior Engineering Manager, Google, United States of America, and Dr. Abhrajyoti Ghosh from Bosch Institute, Kolkata, all participants and my dear colleagues. It is of a great pleasure for me to welcome all of you this webinar on the occasion of National Science Day celebration, which was jointly organized by Maulana Azad College and Microbiology Society of India under the financial support from DBT Star College scheme. 28th February is celebrated as National Science Day throughout India to mark the discovery of the Raman effect. This year, the theme of the National Science Day is the future of STI, a very pertinent theme for today's era. The science, technology, and innovation impacts on education, skill, and work. Like every year, this year also we are going to celebrate this National Science Day through this webinar and an online poster competition for undergraduate and postgraduate students. We have received so far total 46 posters, and those posters are already sent to eminent experts for their evaluation. Result of this winner of this poster will be declared today in college website after 6 p.m. I hope all of us will enjoy these lectures of our two eminent speakers. I wish all the best of this webinar. Now I am going to introduce our two speakers. The first speaker is Dr. Sudhir Shahu, who is a senior engineer manager Google San Francisco, USA since 2018. Previously, he was at YP, the Real Yellow Pages, as Senior Vice President Engineering from 2009 to 2017, as Software Development Engineer at Microsoft from 2007 to 2009. He earned his BTech degree in Computer Science from IIT Delhi and PhD in Computer Science in Duke University. He is expertised in machine learning, algorithms, distributed system. We are really grateful to Dr. Shahu that he gave us consent as a resource person for today's webinar, and he will deliver his lectures on computer science and evolution of interdisciplinary education. So once again, on behalf of college, I send you my warm welcome to Dr. Shahu. And our second speaker, Dr. Abhrajyoti Ghosh, who is an associate professor of Bose Institute uh, at the Department of Biochemistry. He was a Ramanujan Fellow at Bose Institute in 2018, and he was working as a postdoctoral fellow in the Max Planck Institute for Terrestrial Microbiology, Germany, during 2008 to 2012. He is a recipient of many awards and fellowships, some of which are EMBO Travel Grant for workshop at the University of Vienna in 2018, recipient of GST Fast Track Scheme for Young Scientists in 2014, recipient of International Travel Grant from DST for participating Gordon Research Conference on Archives at Italy, recipient of FEMS Congress Grant for Young Scientists, recipient of the Max Planck Fellowship. He has published 41 papers in peer-reviewed journal, written 6 books chapter. His area of research of interest is microbial diversity in stress condition, antibiotic resistance in environment, and many other areas. So with these few words, I also convey my best wishes and warm welcome to Dr. Avrojyoti Ghosh. We hope that from these 2 lectures, we enlightened ourselves and the participants will get updated information regarding these 2 areas. So thank you, sir, both Dr. Shahu and Dr. Ghosh.
Okay, so we will begin our lecture. For the participants, participants can place their questions in the chat box, and this session will be moderated by Dr. Abhishek Mukherjee, Assistant Professor, Zoology, Maulana Azad College. So, Dr. Sahu, you can start your lecture. Thank
Okay.
you, sir.
Can
Participants, please yes, sir. Just some point for the participants. Participants, please pin the screen of the speaker to view uninterruptedly. Thank you.
yeah. So can everybody see my screen?
Yes, it's visible.
Okay. All right. So first of all, thank you
Yes,
very
sir.
much.
Yes.
Okay. So first of all, thank you very much, Maulana Azad College, for inviting me for the National Science Day. It is a great honor to be speaking at such a big occasion, and yeah. So thank you so much, and I'll get started now. And so from this presentation, so it is not very technical. I think my main goal here is to try and essentially convey the message that in recent times there has been a lot of development in computer science. It is touching a lot of aspects, almost all aspects of our lives, and it and that continues to grow. So that is one thing. And then secondly, computer science as a discipline is also starting to influence some of the other disciplines, and that has led to creation of lot of interdisciplinary areas, and there is a lot of progress going on in those areas as well. So like through this presentation, I just want to essentially shed some light on like what are these areas and what are the opportunities there. And
Physics.
hello, sorry.
No, sir. I think it's some problem. You can continue. Sorry, sir.
Okay, no problem. um, all right, so yeah, so with that, yeah, I already mentioned that the main takeaway that I'm hoping to get out of this presentation is that basically there has been a lot of revolution in the computational power over the years, and in terms of the processing power of the computers, and also the internet speeds, and also the devices that are like all pervasive. So because of all of that, we are now starting to tackle problems that I think people used to think about them 20 years back also, but we are not able to solve them. Some of those problems used to be considered ahead of their times in early 2000s, etc. And now actually we are solving them. So that is one thing that's happening, and as a result of that, lot of new opportunities they keep opening up in the core computer science as well as in the emerging areas, which are essentially at the intersection of computer science and foundational sciences. So some example is computational biology, and other example would be biocomputing. And I'll just give little introductions of these through this presentation. So going forward, I want to essentially tell a little story on how we have evolved from, I think, 80s. And I come from Gwalior, MP, and I don't remember seeing a computer in 80s. Actually, I think the first time I saw a computer was in 90s. So some of this is essentially just what I have read on the internet or other places. So based on that, but in 80s, the computers used to be, it was like this precious device that that had to be kept in an AC room. You had to remove shoes before entering because it cannot handle any dust. And this is how it looked like. Um, the first evolution in this area happened in 1990s, and I think the big thing here was Windows 95. So when that came, the computers suddenly became much more user friendly and fun. They also started becoming visible in more schools and offices, etc. So as a device, essentially the accessibility of computers increased, the reach increased, and I think that was really a pivotal movement. Then I think the another event that happened in 90s was internet was starting to spread. If people remember the dial-up connection from that time. Um, so again, I think even though it is kind of unimaginable now how slow it was, that was a very big leap at that time. And if yeah, and actually one of the first usage of internet I remember was the train reservation system, and I think it was something that was active in late 90s also, because I remember buying tickets from Gwalior to Delhi where my college was, and I do remember I think the computerized ticket system at that time. So this was, I think, the first application of internet that I remember seeing. Yeah, then the another one that some of you might remember, the email. So that was the new thing in late 90s. Hotmail was the I think the big name at that time in the world of emails. Uh, Another, I think, device, computerized device from that time was ATM. So that's all of these were pivotal. Like this is what shaped the decades to come, and these were like each of these were big, I think, leaps in their own right. Then I think 2000s were just remarkable, and I think the biggest credit goes to broadband and Wi-Fi. So essentially, when broadband, the internet speeds increased and it became more accessible through Wi-Fi, then all of a sudden, lot of uh, once the accessibility to internet increased, then I think online became a thing. And at that time, I think some of these companies that had really born in late 90s. So for example, Google was born in 98, Amazon was born in 96, Expedia was also born in 96, for example. But all of these, they really started thriving once the internet became uh, a more accessible, more I think common technology. Then, like fast forward 10 years, again, I think the mobile was the big invention. I would say iPhone, I think, really changed the scenario there. And yeah, everybody can remember, can see the social media. I'm showing a Facebook screen on the mobile phone. So with the I think I would say early 2010 was when I think the all of these social media platforms, they I think started becoming more and more available in 2010s. The apps became much more user friendly. Uber was a small startup in I think 2009 or so, then it started becoming more. It started spreading, expanding in more newer areas, and then I would say around 2015 or so the technology called blockchain started becoming more and more popular. And some of you might know about blockchain. So essentially, this is the technology that powers Bitcoin, which is a lot in news these days. So blockchain is the underlying the technology there. So yeah, so these were the things that happened in 2010s. We are just getting started in 2020s. I think the first thing that we experienced was COVID-19, and I think it was amazing that because of the technology that is available to us, that even through lockdowns, there were a lot of activities that could still continue like before. So, for example, the presentation that we are having right now. So it is all a result of the advances in the video technology. Like 10 years back, it was very difficult to get a video of this quality, like halfway across the world, streaming at full bandwidth. So all of these gradual advances in technology, I think they actually made it a little bit easier to continue through some of the difficulties. Again, I mean, I I think the COVID-19 has been a very tough time for um, everybody. But I would say that uh, I think something like this would have been harder 20 years back. So on that note, I think there is a lot more that would happen in next 10 years, and at this moment it's a clean slate. So I want to talk about how to become part of that bigger revolution that is going to happen over next few decades in the field of technology. And in my opinion, I think there are many ways, but I would talk about three ways. So one of them is the core computer science. And again, my focus, I have all my life, I have only done computer science. So I'm naturally biased towards it. So I'm just going to talk about that. Uh, but yeah, so core computer science is like one of the ways. Then other is interdisciplinary computational sciences, and examples would be the computational biology, computational economics. And then I think there is another, I would say, pillar or another direction in the area of computer science, which is essentially about the fundamental shifts in how the computation is done. And that is where technologies like biocomputation, nanorobotics, or quantum computing fall in. So I want to like talk about maybe one example from each of these areas. Um, and I don't know how much time I have. I think maybe another 20 minutes or so. So,
Yeah, you continue.
okay, okay. So yeah, so first core computer science. I think this is probably nothing new here. I mean, it is kind of something that I think a lot of us know, that in core computer science, software engineering is one of the very popular disciplines today. So the software engineers, they write code, they essentially tell the computer on what problem they want to solve. So they need to give very clear instructions to the computer, and then the computer just follows all the command. So software engineering is essentially just an art to make yourself better at writing those commands to the computer. Yeah, so like this quote here says, so coding is about solving problems. That's the essence of writing computer programs. They should solve problems for us. So that's the end goal. And I have given an example here, just a toy example of, for example, if somebody wants to calculate the average of some numbers, then essentially this is how you can write it. And basically the computer will understand this language and it will do the job for you. So this is just an example, like whatever job you want computer to do, you can just write it in the form of a program and it will do that for you. This was just a toy example, 10 lines. Generally, like if you think of anything that is that we use in real life, like consider Uber app, that probably contains millions of lines of code. Or Facebook, I would say hundreds of millions of lines of code, and Google the same way. Uh, yeah. So going forward, if people want to uh, figure out a career in software engineering, so there are really three three pillars there, really, like only three aspects. So one is the programming, and for that I think there are common languages like C++, Java, and there are a lot of other popular languages also. So like prospective uh, candidates, they can essentially pick any of the language that they like. C++ and Java are more popular, but I think the main thing is to just learn the art of programming. Then I think the other two dimensions or other two aspects would be to master algorithms and data structures. So this is essentially really the meat of creating solving problems and giving instructions to the computers. So algorithms are the way you will want to solve a problem, and data structures are essentially the uh, the structures in which you store the intermediate data, which your algorithm uses. And then the third thing is databases. So database, a simple way to think about database is that it is just a store of information. So your algorithms or your program, it is talking to the databases all the time. And as a software engineer, if you can get the mastery in these three areas, programming and algorithms and data structures and databases, then I think 90% of the problems you can solve. These three things, uh, they'll cover 90% of the software engineering. So now moving on, uh, I mean, we are living at a actually pretty good time in terms of learning. So it has never been easier to learn a new technology or actually learn anything new. So these days, if people want to learn dancing, they can, like I personally know people who watch YouTube videos and then they practice dance while looking at those. There are people who are learning to cook uh, by watching videos or by following recipes on the internet. So same applies to programming and algorithms and databases that I talked about. It is super easy these days, and all the resources are there available, and most of them are also free. Online tutorials, there are some sites, and there are many more. I just gave you three examples of these. They are available. I mean, books obviously are available. People, you can order online. They'll just get delivered to your house. And there is something called MOOCs, so massive open online courses. So these are slightly better than books and online tutorials, because here it is actually a real lecture that you can participate in. There is actually real homework that you can do. The big convenience is that you can do it at your own leisure. So lecture, you don't have to attend live, it's all recorded, so you can just listen it however you want. And generally the homework or the assignments here would be something that could be graded by the computer itself. So as you finish your homework, you can submit it, and the site will, or the software will actually grade it for you. So MOOCs are actually very, I think that is completely revolutionary idea. I think that is taking education far and beyond. Right now, I think MOOCs are used mostly in the computer science fields, but gradually, I think other areas are also going to start adopting them, given how useful they are. Now, I think one important thing I do want to call out is that learning theory is one thing. I think you really learn it when you actually practice. So in CS, the project-based learning is something that is highly encouraged. And like I mentioned earlier also, that programming is not just about learning a language, it is about abilities to solve the problem, and practice, practice, and practice. So those are the three principles you need to follow. The resources are all available, and learning, and it can be done at any time. So for me, I saw the computer. I mean, I saw it before, but I used the computer for the first time when I went to college. So yeah, so it is actually never too late to learn software engineering. Then I think sometimes I get asked a question that, okay, what you're talking about is good for the generalized software engineer. What about the specialized software engineers? So for example, there is a very hot field these days called machine learning engineering, and mobile app programming, or game developers. So what about them? So again, I think resources for them are also available, all online. The learning principles still stay the same. Project-based learning is the way to go. Practice, practice, practice is the way to go. Uh, Going forward, I think one other question that I get asked sometimes is, okay, what is the difference between ML engineer versus data scientist versus data engineer? And honestly, the answer is that they all have a lot of overlap. I mean, there are all these interesting Venn diagrams that I found. They kind of show that, if you take these three fields, computer science, maths, and then domain expertise, then essentially the combination of all three is data science, and computer science and math is machine learning, and essentially doing only the data engineering is minus, I don't know, sometimes you can actually do data science as well. But the whole point is that I think some of it is artificial, I think, distinction. uh, Technically they are all very similar, and there are some nuances, but essentially you can learn one and then you end up learning a little bit of others also, and then I think as your work needs, you can start diving into specific areas. And so I have seen a lot of people who are machine learning engineers, they actually are also good data scientists, and vice versa. Similarly, good data scientists end up being good data engineers also. So all of these distinctions are there, and these questions do come up, but there are only subtle and surface, superficial differences. That is my opinion. Um, and moving forward, okay, so there was a lot about the software engineering, the first pillar that I am most familiar with, but I do want to touch upon the whole interdisciplinary aspect. So we can talk about, I think we saw in the okay, so in terms of in the interdisciplinary computer sciences, I think the, sorry, some question, okay. Yeah, so computational biology, computational physics, and actually many variations of computational sciences have uh, recently come up. And the biggest reason is that the computational powers have increased over the years, that distributed computing have become possible. So a lot of problems that used to be solved just by a mathematical model or a theoretical proof, now actually it is possible to run simulations for them, or be able to run computations that were earlier just considered very difficult to do. So that is really the reason why uh, a lot of foundational sciences uh, have basically emerged with this computational science aspect. And yeah, so the paradigm is really, I think we have seen this similar graph earlier also, that you have computer science, and then you have the science discipline, and then you have the applied math. And like wherever the three of them meet, that is what becomes the computational science aspect of it. And some examples are computational genomics, computational biology, computational finance, economics, neuroscience, and there were many more. But again, I think the goal here is to just showcase to the audience that it does not matter which field you are in. there is probably a big computational aspect to that field which is starting to emerge. And if basically that is where your interest lies, then there are a lot of opportunities in that area also. So that is really the message I want to convey. And all of these are just examples. Yeah. Oh, yeah. So I also actually had remembered one one motivating problem for computational biology, and I am not sure if uh, people are familiar with it. But uh, so, for example, you are given two sequences. So assume that these are two DNA sequences, and then the question that is asked is, okay, from first sequence to second sequence, how many minimum number of mutations you can reach? And the algorithm to solve this problem, it was designed using something called dynamic programming. And I think this essentially forms the basis of a lot of the computational tools that are used in the field of computational biology and computational dynamics. But there are, again, the idea is to essentially showcase that there are very algorithmic problems in the field of biology, and that is where this intersection of computer science with biology can actually help solve things that are otherwise harder, much harder to do. Okay, moving on. Yeah, so now this brings me to the third section of the talk, and here I want to talk about two areas which are essentially fundamentally changing the way the computation is done. So first one is biocomputation, and then second is nanorobotics. So I'll talk a little bit about them. So let's start with biocomputing or DNA computing. So here the idea is different. So it is, in some ways, it is reverse of the computational sciences that I just talked about. In this area, we are saying that use the biology or the ideas of biology to solve a computer science problem. In other one, we were talking about taking computer science and solving bio problems. Here it's reverse. And there is also a very, I think, very nice example of a problem known as Hamiltonian path problem. So it's a very common problem, but it is quite difficult in CS. And I'll just give a very high level idea of the problem itself. So see, there are four cities here. So Boston, Chicago, Detroit, and Atlanta. And these arrows show a flight from one city to other city. Then the Hamiltonian path problem is basically saying that can you like start from one city and essentially touch every city once and uh, and be able to reach all the cities. So does such a path exist in this graph or not? So that is the problem. So in this particular graph, so Atlanta to, so for example, I think you can go to, um, yeah, so I think you go from Atlanta to Boston, Boston to Chicago, Chicago to Detroit. So this is one Hamiltonian path. You can follow this path and you can touch every city exactly once. Right. So this is the problem is that given any graph, does such a path exist or not? And there is a scientist named Leonard Edelman, who is known as the father of DNA computing. He first thought of solving this very hardcore computer science problem using DNA computing. And then what he did was he just took the cities and he encoded them in a DNA sequence. And then he created an encoding for their flights. And then essentially he put all of them in a solution, mixed them in a test tube, and along with the ligase enzyme. And then he let this thing run. And whatever is the longest molecule that get formed, that is really the answer to the problem. So this way, like he used the ideas of biology, the ligase enzyme, the I think the hybridization concept of hybridization itself, and use those biological processes to solve a computer science problem. And this was done in, I think, 1994. And after that, this is how this field of DNA computing was born. then actually there were scientists in I think Canada, in Calgary, University of Calgary, who actually created the entire uh, mathematics out of DNA computing. And yeah, so I won't go into details of that, but this was one example of how I think bringing two disciplines together, you are able to solve things that are otherwise not possible. So another example in this area, yeah, it is actually quite a fascinating example. Is so you are seeing some shapes on my screen, and all of these are actually built out of DNA nanotechnology. So, for example, this smiley or this octahedron or this wireframe structure. So all of these were like you take some DNA sequences, independent DNA sequences, and you essentially allow them to react with each other. But you have chosen the sequences in a way that specific shapes are formed, specific areas of the strand attach with specific area of other strand, and they all combine together to create this shape. So this was actually quite revolutionary in the area, because if you think about like creating really nano scale structures using the traditional tool, it is going to be very, very difficult, because we are talking about nanometer. So I think if the usual computer chips, they are usually at the micron level, micrometer level. So at that time you can use technologies like lithography, etc., which are more top down to create some tools. But when we are operating at nanometer scale, then the top down will not work. So in this whole area of DNA self assembly, you are actually going bottom up. So you have the molecules, you have the sequences that you want, and then you are letting them self assemble. But you have chosen the sequences programmatically in a way that they only combine in certain ways and create certain structures. And people are able to create all of these. I think they are probably like 100 nanometer by 100 nanometer, really, really tiny structures. And you can create billions and trillions of these in a matter of few hours. So it actually is able to generate material in a specific shape and form at a scale that was never seen before, right? Trillions of smiley faces. It's not possible to create them in any factory at such a big scale. So this again, nanofabrication is a big area where people are trying to create these structures, and then at the end of the day, they want these structures to do certain things. So one of the idea that used to be discussed a lot when I was doing my PhD was if you can create this kind of a cube, a box, which can open and close, and then you put the medicine inside it, and then this cube reaches the can basically where the disease is, and it opens right then and there, and the medicine could be delivered to the targeted place. So that would be something that would be really revolutionary in the area of medicine. And actually, there was one experiment. Oh, yeah, nano machines. So uh, yeah. So again, I mean, I've already talked a little bit about this, but here the idea is again, use the chemical reactions in a way that your nano structures, they are able to perform certain motions. So here this is actually a walker. So if you see that this is the cargo, this is a walker, and this is the DNA road, and it is walking from here, moving forward. And again, it is doing it through a combination of hybridization and restriction enzyme reactions. And I think this was I think a vision that you want to create a robot that could just walk on a microtubule and do things in a certain programmatic controlled way. And I'm actually just moving on to the experiment, a study that was done in 2018, which actually talks about creating this DNA origami, this DNA flat sheet, and attaching two proteins. So thrombin protein here, then again, because of the self-assembly, it rolls and it creates this tube. Then I think they were able to inject it to the arteries, and it reached the tumor, I think there were certain enzymes, I think there was an aptamer sequence here that could detect the tumor, and as a result of that, when this reaches the tumor, it actually opens up, and then the thrombin protein is able to create clots here, which reduces the blood supply to the tumor, and the tumor reduces in size. So again, this was a study that was done in mice, and it showed some promising results, and there are practical limitations of it. But I mean, this shows that, in 10 years time, something that we used to just talk about, that, oh, we will have this kind of a box which will deliver the medicines in a targeted way, somebody was actually able to run an experiment, a real experiment, doing that. So the science and technology is moving at a really fast pace, and I think there are opportunities, new opportunities are opening up as more and more areas are essentially joining forces, and a lot of cross learning is taking place. So with that, I think I come to the end of my presentation, um, and
Thank,
open for any questions.
thank you, sir. So much complexities and creativity was there in the talk. So much is happening with computer programming this day. Very, very innovative. Very, very, very complex. I find it very complex. Anyways, Dr. Abhishek Mukherjee, uh, are there any questions?
Yes, thank you, Dr. Sahu, for your exciting presentation. So only one question from Dr. Swapna Mukherjee has been posted here in the chat box, that she wants to ask you about that. I would like to know your opinion about the AR and VR engineering, which is coming up with a huge prospect. Sir, please.
Yeah, so I think that is a great question, and that is a big area of future innovation. I think um, so. Right now, we are at a very early stage in AR and VR. But I think that day is not far when we will see much more application of it. I think COVID is probably going to expedite some of those innovations, because right now there is really a need of uh, like being present somewhere else in a virtual way. I mean, the Meet and Google Hangout and Zoom, they are doing something, but it's not as real. But if we could have good virtual reality, I think it would be much better experience. So I also know of some. There are certain companies that are actually trying to use these tools for things like, when people want to buy a house and they want to tour the house, then you should be able to do it through AR, VR technology. So I think that is really coming up, and it will happen sooner than we think. There are definitely many positives, which essentially it removes the distance from the picture. If AR and VR could be done properly, then I think the traffic can decrease a lot. Like the meeting that we are having right now, I mean, this could feel like a real meeting, a real, in-person meeting, if there was if when the AR and VR technology is there. So it could solve a lot of problems. I think in my opinion, it definitely solves the distance problem. And again, my views are very biased because of that is the problem that I see. I live here, my parents live in India, and it would be nice if uh, we could feel that we are in the same room through a technology like this. So yeah, I think it will happen sooner than we think. Uh, and okay, next question: how AI is shaping all over the science world? Again, I think AI is starting to do things that earlier were thought to be very difficult. So I'll give you some examples where actually people are able to resolve a lot of the simple complaints through AI. Where so if you remember talking to let's say customer service, where you are put on hold for 30 minutes, right? Instead of that, if there is AI, it is like you don't have to wait. So it solves some of that problem. And uh, I mean, again, AI is everywhere. There are challenges. I think it has to be done in a very responsible manner. I think with any new technology, there are always going to be certain dangers which only become, I think sometimes it is very difficult to see them earlier. So I think that is something that we have to keep in mind. But uh, again, AI is something that is already starting to power a lot of things. I mean, if you think about the driverless cars, so that is something that is actually happening. Many car companies, they are uh, really trying to make a lot of progress in that, and uh, that will be a phenomenal example of AI in the real world. Uh, Again, I think that solves a lot of problems. And that might change a lot of things, the way things are done today. I mean, yeah. So sometimes these technologies are going to be so disruptive that it is even very difficult to imagine the type of things that they they will change. And I think that both AR, VR, and AI, they fall in that bucket. Yeah, I so the
Next question is also about AI.
so last yeah so uh, yeah I mean, again, I think that is why we need to do AI in a responsible way. I do think that, I mean, movies are movies, and some they are far fetched and exaggerated, but um, like just like knowing about the state of art today, I think it is very, very far away to have an AI that is that smart. But it also means that we just need to be careful and thoughtful when as we are like doing new innovations.
Sir, I have a question. Don't you think that with all this AI technology, human employment will decrease? And so, what you have to say about that? Because population is rising, and if technology, definitely technology is progressing, but if with that we cannot employ more and more people, then I think that will be a serious issue for the world.
Yes, I think that is true, and that is why I think people smarter than me should. I think they are. I think thinking about all of those aspects as well. But that is valid. That is a real uh, discussion, real question that come up when we are thinking about automating something. That will it take the employment away, or so again, I think there is a very fine line. I think there are certain things that bring convenience, and I think if you take it too far, then I think it can uh, have that damaging effect that you're talking about. So yeah, I don't have a great answer for it. I think it is it's a real thing, and I think whenever we go there, we will have to be careful. And
Yes, sir, but these days the trend is that we feel very excited to take delivery from a robot than from a people. I mean, that's what I feel when I see the social media and everything. Everybody is so excited. They always display that yeah, everything is happening with AI technology and no humans are employed. So I think that is going to take over the world, the AI, and I think this is going to create a really great pressure for the employment of the human. Anyways, thank
no,
you.
I think that's a very important thought. And yeah, uh, I can't argue against that. That's a
Okay, sir. But
thank
yeah,
you.
yes, sir. Thank you.
Okay,
Okay.
I will stop here.
Yes, sir. It was very nice to hear from the resource person. So our next speaker is Dr. Obrajyoti Ghosh. Sir,
Yeah,
please start your session.
okay. Thank you.
Welcome, sir.
Am I visible? The presentation?
Yes,
Yes,
sir.
yes, yes,
Yes,
yes.
sir. It's visible.
Okay. First of all, thanks to the organizer for considering me to present for today's event. I remember 2 years back at Bose Institute, I actually organized this event myself. I would also like to thank Dr. Shahu for vivid presentation on computer science and its relationship with biology, their cross connection. And indeed, we do use a lot of computer science in our to solve our biological problems. I would also like to thank Professor Mukherjee for delivering a nice introductory remark on the event, and also Professor Dasgupta for introducing both of us. Well, for the sake of the story, I will show some of our results, and I would like to tell the students especially that don't go with the results, just try to understand the theme or the flavor that we I'm trying to give you. Okay, so the title of today's topic is Sundarban Mangrove Ecosystem, a Microbial Treasure Trove. So I am from Bosch Institute. I am from the Biochemistry Department. I hope all of you know this is our century-old building, which is the
Visible
main.
কোথায়? আমি তো পাচ্ছি না। Oh,
Sorry.
দুটো ভিডিও না?
Shubhir sir, your microphone is on. Sorry, sir. Sorry for the interruption.
Okay. So this is the main campus of Bosch Institute, which is situated in Raja Bazar, and this is actually Acharya J.C. Bose's residence. It's, and now it is our main campus. We have a beautiful museum in there. So I would like to invite all of our students to visit that museum and to motivate yourselves by visiting there. And then we have actually many campuses. This is another campus, which is the Centenary campus where I do work, and this is our new unified campus at the New Town. So this is where I belong, and we do different things. And among different things, this is one of the aspect that I am interested in: mangrove forest, and in mangrove forest, how microbes play important role in keeping the sustainability of mangrove forest. And our model mangrove forest is Sundarban. So in general, mangroves are located along the tropical and subtropical coastline around the world. As you can see, all these green regions are the mangrove forests, and they serve mostly as means of livelihood for coastal population. They provide food in the many foods like fishes and so on. And also another thing, they protect the coast from tsunamis and cyclones. By now we all know, and more we intervene into such forests, actually we will destroy those mangrove forests, and that will actually make our lands more vulnerable for with respect to repeated cyclones and tsunamis. These things actually we all know. Now, why they are so important? They are hotspots, especially if you talk about Sundarban. Sundarban is one of the official hotspot of microbial and flora faunal diversity. So normally mangroves are having lot of different types of flora as well as different types of fauna. And we know from our Sundarban that they actually have lot of different kinds of mangrove plants and also different types of animals that I'm coming shortly. So this is our Sundarban, which is the largest tidal halophytic mangrove forest in the world. Not only that, this is a World Heritage Site, UNESCO World Heritage Site, and it is being shared between India and Bangladesh. 60% portion is in Bangladesh and the 40% is in India. And it is a very famous tourist spot with respect to the fact that it is the home for Royal Bengal tiger. And also the name comes from a mangrove plant called Sundari tree. The interesting fun fact is Sundari is actually one of the rarest mangrove species one can spot in Sundarban, because with the increasing human intervention in this mangrove environment, we are actually seeing a change in its flora as well as fauna. And when if you try to spot a Sundari plant in Sundarban, you may find it difficult to spot one, because the entire vegetation is actually shifting due to human intervention. I will be coming to that later. Okay, so as I am a microbial biochemist, and my own interest is microbes, so we normally look into the microbe world of Sundarban. And in Sundarban, it has very unique physical features, like different types of physical factors like salinity, and also oxygen concentration on the sediment is less because of regular inundation. And also the geographical location is so unique that you can see lot of differences in different parts of Sundarban, and it is rich in nutrients, and that makes it highly productive ecosystem. Actually, mangroves are among the most highly productive ecosystems around the world. So there are different types of microorganisms, diverse microbial communities one can find in any mangrove, including Sundarbans. And also these microbes are actually playing vital role in nutrient cycling. So from our school days, we have been learning about different types of nutrient cycles, and those nutrient cycles are controlled by different types of microbes. We study also during our bachelor's, even in master's, that microbes play important roles. And in environment like Sundarban, where you have little chance to, I mean, cope with varied environment, these microbes play essential role in actually let allow different types of pollutants to go into this mangrove ecosystem. And under such environment, microbes also play important role in degradation of different types of waste by the means of bioremediation. And that is one of the topics many people are trying to understand, how microbes actually adapt, cope with such environment with increasing concentration of different types of waste, and how those microbes are capable of remediating the waste materials, and thereby keeping the environment sustainable. This is one of the area where many people are working around the world, and also on Sundarban, in Bakipur, in Odisha, some regions, also in Kerala, there are people working on how uh, the waste disposal, the pollution level is increasing in all these mangrove uh, environment, and how microbes are actually coping with this increasing concentration of different pollutants. So also these environments are rich source of novel compounds such as antibiotics, because nowadays, as you know, antibiotic resistance is one of the major concern in medical practice. And researchers are looking for more and more new antibiotics. Now, mangrove harbors different types of microbes, and those microbes actually uh, provide new source of different types of compounds in the form of antibiotics. Although one thing I would like to mention, that microbes do not produce antibiotics for us. They do produce antibiotics for their own survival in natural environment, and we actually identify those molecules produced by certain type of microbes and use them in the form of antibiotic for our own purpose. So if you talk about micro inhabitants of Sundarban, there are like different types of organisms that people study. Bacteria, fungi, actinomycetes, archaea, and cyanobacteria. So these are 5 different types of major micro-inhabitants that are present in Sundarban. And so, like many other researchers, we also work on many of these different types of microbes. Now, why microbial research is important? Why mangrove microbial community research is important? Because, so suppose this is basically a natural environment, natural mangrove environment. Now, human intervention can lead in deterioration of this mangrove environment to poor condition and finally a degraded environment. And this degradation is assisted by poor nutrient and carbon cycling because of loss of microbial communities in this mangrove. And also you are actually by adding, for example, pollution to any given mangrove ecosystem, you are actually disturbing the microbial diversity in that given environment or ecosystem. That ultimately leads to degradation of the environment. Now what researchers do, they actually look into different stages of degraded mangroves, and then they collect samples from all these different types of mangroves, and then they perform lab-based studies with culture collection, or even nowadays using next high throughput technologies. They do lab-based studies where they don't go for culturing microbes, but they directly study the DNA and RNA content of a given sediment and try to understand how microbial communities are actually changing over the time with addition of different types of pollutants. Finally, once they understand the microbial communities in a given mangrove sediment, which is the natural microbial communities, they try to actually mimic such microbial communities in degraded mangrove environment, and which is by the means of microbial intervention, and thereby trying to actually restore the mangrove ecosystem. So this is in general a holistic view of how researchers look into the microbial research priorities in mangroves. And this is one of the very recent publication, as you can see. The interesting thing is characterizing mangroves, microbes across the scales in a changing world. So the changing world is very unique and important. And I took this slide for my presentation because I would like to give you a flavor that what we do in Sundarban is somewhat like this. So we try to look into Sundarban, different parts of Sundarban, in a way that we have a gradient of pollution in Sundarban also. And we would like to know, with that increasing pollution, how the microbial world is changing and how such a change is actually leading to a change in the habitat or the flora and fauna with which microbes are deeply associated. Okay, so from researchers' perspective, there are 3 issues that normally molecular biologists or microbiologists ask. So the first question they normally ask, who are there? That is very important, because when you are trying to look into an environment, you need a control always. So the microbial communities present in a given environment over a time period, and also the metadata that we call it, it's the pollution status of that given environment during that time period, is going to tell you how the microbial world is changing. So who are there is the basic and very primitive question. Then what are they doing? So some organisms are there in any given environment. So if you go to your the field next to your home, you will see there are bacteria, archaea, fungi present in that soil too. So the question is, what are they doing? Are they helping the environment to sustain, or they are trying to cope, or what are they doing? That is the very basic question. And finally, how do they survive and interact? That is the third question, which is very important, because when we work or study microbes, we study like single microbe. Means when we study about Escherichia coli, we talk about Escherichia coli, its different properties, its genetics, so on and so forth. But in nature, they do not live alone. They live with other organisms and also other flora and fauna in association to other flora and fauna. So it is very important to understand how do these microbes in nature do survive along with other organisms, and how they survive with the flora and fauna of a particular habitat. So for every each of these questions, we have different types of approach that we take. The first who are there in 1998, Joe Handelsman actually proposed a name called metagenomics. This is what uh, microbiologists nowadays try to adapt in order to understand the microbial communities in a given habitat. The main reason is that up to 1 to 3% of microbes in any environment uh, is only cultivable. The rest is not cultivable. So that is one of the major reasons why people for a long time have been trying to develop a technique where they can look into microbial communities without cultivating them. So metagenomics is one of the solutions to that problem. Nowadays, it was 1998. It has been evolved so much nowadays. You can really look into microbial communities within a few hours with the advent of high throughput techniques like next generation sequencing as well as computational biology. So what are they doing? If you want to know what are they doing, you have to isolate, characterize, and also you can do functional gene diversity, which involves again high throughput technologies to understand what are the genes present in a given environment, and also in that environment how these genes are actually being expressed, so on and so forth. Finally, to understand how do they interact and survive in any given environment, you need to perform functional genomics, which involves different types of techniques like transcriptome, also proteomics, so on and so forth. So what we do in Sundarban, mostly we try to catalog the microbial diversity, community diversity and distribution in Sundarban. Also, we are interested to assess the impact of climatic and anthropogenic stresses on biodiversity, and finally to explore microbial function and study beneficial microbes from Sundarban. For your information, it's almost 10 years, until 10 years before, we had no information about the microbial community diversity in Sundarban. It has only evolved since 2010, and now we at least understand a portion of it, and I believe many of you will join research after completion of your masters and will contribute to understand better the how microbes play role, even the flora fauna play role in Sundarban for to maintain its sustainability, because mangroves are very special. They too actually help us a lot by protecting the coastline, which is very important. And all the organisms whichever present in those environments are essentially contributing in that process. Okay, so in general, we work on sediment soil or the bulk soil and the rhizosphere soil. We try to understand how bacteria and archaeal communities are distributed, how their distribution is in those two different types of soils. Also, we perform culture-based analysis, and then we try to understand how they contribute in maintenance of that ecosystem. So this is in a nutshell what we do at Bosch Institute, and also with some collaborators in different institutes. And before we go into the detail, So we need to understand the strategies for microbial diversity assessment, what is called metagenomics in this case. So the idea is either you can, from your syllabus or from your studies, you already by now know that there are some marker genes, for example, 16S are RNA genes that we use to identify a given microbial species. And there are two approaches that one can take. One is the clone-based assessment using capillary sequencing. Capillary sequencing we all do for routine sequencing. Here you cannot sequence a lot of DNA sequence at the same time, but the quality of sequence will be higher. And so what we do, we first take the soil, total soil from any given environment, then we isolate the total DNA, and then we amplify the 16S rRNA gene sequence by performing polymerase chain reaction, and then we clone that sequence amplified fragment of DNA into different types of vectors. There are different types of vectors available, and then we go through one round of passage through E. coli and then isolate each of these plasmid DNA, which are recombinant plasmids, and then we individually sequence each of these fragments cloned into the plasmid and thereby identify on the basis of sequence DNA the organism. So this has been in place for that, let's say 95 till 2005. So people used to do sequencing with the help of clone library based methodologies. From 2006 on, with the advent of next generation sequencing, everything has changed. And it is still evolving. It is a very new area of research, not only in metagenomics, but also in transcriptomics, in chip sequencing, and there are different types of sequencing technologies that is beyond the scope of today's presentation, but you can use different types of techniques to understand how next-gen sequencing platform is useful. So in this technique, what you do, you take the soil, you isolate the total soil DNA, and that total soil DNA, you can either go for amplification, which you can amplify the 16S RNA gene sequence, for example, like this, with addition of some adapters. Don't go into the details. Then you can directly sequence without cloning them, because cloning takes a lot of time and you have to sequence individual clone. But if you want to go for next generation sequencing platform, you can directly sequence a lot of DNA at the same time using these instruments, and your data will be at least 100 to 1,000-fold higher compared to the capillary sequencing platform. Means you will get a lot of data. So when you say something from a small cohort, for example, like 100 clone-based assessment, clone-based sequencing, you can tell a story. But when you have 10,000 similar sequence available from a given environment, it is statistically better. So people used to perform application sequencing based analysis using next generation platform nowadays. Let me tell you one interesting fact is that it's all started with discovery or marketing of Roche 454 pyro sequencer, which was the first next-gen sequencer came into the picture in 2006. And let me tell you, in 2016, they have stopped making new instrument because there are other instruments already available and cheaper, cost effective than 454 pyro sequencing. So see, this is really an evolving area where people are contributing, and we are getting lots of different types of information by doing different types of sequencing. I am talking about only metagenomics. So don't go into the data, just the theme. So the thing is, this is just a data showing how the bacterial community is distributed in Sundarban sediment. So there are different key findings that we had at that time. It was old. So dominance of proteobacteria in the sediment, which is kind of very common. Proteobacteria is one of the very dominant member in any given soil sediments. Also in Sundarban, we see the same. Any deviation from this observation? Well, let me give you another example. Like if you go to the northern part of this state, near Darjeeling or so, if you analyze the soil in Darjeeling or in that region only, you will see it's not dominance of proteobacteria. It's a dominance of actinobacteria, and that is markedly different from. So these two soils are completely different. In this case, it is proteobacteria which is dominating the sediment. Not only that, the surface sediment, so we have performed sequencing at the surface and also in different layers. We could see that surface sediment in general much more diverse, and most importantly, surface sediment is dominated by anaerobic microbes. So this at that time was a big information for us because it was never been studied. So we looked into the microbes and we suddenly found it's all delta proteobacteria, and most of the members of delta proteobacteria are highly anaerobic in nature. So, which gives us a hint that the surface sediment is anaerobic. Now, we also looked into the archaea present in this sediment. Again, don't go into the detail, only this section. So, what we could see, there is a difference. So, we tried to look into the pattern archaeal community in different parts of Sundarban with different history of anthropogenic pollution. So here anthropogenic pollution includes measurement of heavy metals and polyaromatic hydrocarbons. And what we could see that stations where we have less pollution or less heavy metal and polyaromatic hydrocarbon, there we have less or the different microbial diversity or archaeal diversity pattern compared to stations where we have more pollution load. So from that finding, we could actually conclude that haloarchaea, these are haloarchaeal species, different haloarchaeal species. Haloarchaea dominates the sediments with higher concentration of heavy metals and polyaromatic hydrocarbons. At the time when we published this research, we were not aware of any observation similar to this, but there were reports that haloarchaea are capable of degrading polyaromatic hydrocarbons. But at that time, in Sundarban especially, or even in any of the Indian mangrove, we had no such data. So we were confused. Now, when we tried to tell this story to international people, they told us something very interesting. They told us, so you did metagenomics. So in metagenomics, you isolate the DNA and then you sequence it. When you sequence the DNA, you are actually looking into the live cells as well as the dead cells. Okay, so you don't know what percentage of cells are living and what percentage of cells are dead. So if you want to look or find the real picture in this sediment, you have to sequence the RNA. So then we started planning for metatranscriptome analysis, where we have isolated the RNA, we converted the RNA into cDNA, and then we sequenced the cDNA. And thereby we looked into the only to the live cells. And to our, this was our planning, how we performed. So we took two approaches. One is culture dependent, where we tried to isolate haloarchaea, and we tried to characterize them with respect to their abilities to handle heavy metal and polyaromatic hydrocarbons. And then on the other hand, the soil RNA was isolated, converted to cDNA, and performed sequencing to understand how the live cells, archaeal cells, are distributed in the sediment of Sundarban. So when we looked into the metabolically active, so it's called, the live ones are called metabolically active organisms. So when we looked into metabolically active haloarchaea, we found, so again, don't go into the data, just these two. So the halobacteria, which is the representative one, G1 and G2 are the most polluted sampling stations, and there we could see dominance of this halobacteria, indicating that in polluted sediments, again, when you looked into the metabolically active ones, you can see the similar trend what you could see in the DNA. So we concluded from this, pollution saves the microbial diversity and distribution in the mangrove sediment of Sundarban. More importantly, this is the first indication that we got that pollution actually alters the microbial diversity. In general, haloarchaea are the member of Euryarchaeota, which is one of the specific type of archaea, and they are present in different types of mangrove environment, but they are not the abundant one. But interestingly, in Sundarban, we could see in the polluted regions of Sundarban, we have higher concentration of those haloarchaea, which indicates that there is a direct correlation between the pollution status of a given sediment and the abundance of haloarchaea in that sediment. Also, we could see that restoration efforts have shown to recover microbial community distribution in this ecosystem. So what we did, we looked into regions where from government we have restoration effort ongoing. So we took samples from those areas and we measured the content or the presence, abundance of haloarchaea by real-time PCR techniques, and then we could show or we could find that basically in restoration area where the restoration is ongoing, you can see again the microbiota is shifting towards the normal microbiota. So it's no longer dominated by haloarchaea. Now the question is why haloarchaea is dominating. So to understand that, we have started isolating haloarchaea in pure culture. Let me tell you, it's not E. coli or any other bacteria. Archaea are sometimes very difficult to cultivate in the laboratory, and we are lucky enough that we could isolate some of the haloarchaea. Like yeast, you have to give them even vitamin mixture and many other nutrients, because they are not like bacteria. You just grow it in LB and you will successfully isolate some. It's not like that. So it took us some few years actually to isolate some of these haloarchaea, and what we could see that we could isolate many different. Actually, now we have 11 strains in our lab which are completely different from each other from Sundarban. But not only that, we could show that they can use all these organisms can use para-hydroxybenzoic acid, which is one of the major pollutants, and benzoic acids, also naphthalene, etc., where they can use these molecules as their carbon source, sole carbon source. So that indicates that haloarchaea can actually capable of utilizing para-hydroxybenzoic acid or benzoic acid or even naphthalene, confirming that they are capable of handling or metabolizing hydrocarbons in natural environment. So what is its application? How you can apply? Because when we do basic research nowadays, the government is so strict that they always ask whether our research has any implication in or any application. So we actually looked into the possible applications, and we could show that chemical oxygen demand reduction of polluted Sundarban waters was achieved by addition of haloarchaea. So here you can see again, no data, don't go into the data. So we have mostly some haloadaptators and haloferous species, and we could see that polluted waters collected from Sundarban, where we have a lot of actually hydrocarbon particles, because hydrocarbons are hydrophobic mostly in nature. They don't love to stay in water. But in Sundarban, if you go in Sundarban, if you look into the water, it is not clear. It is a lot of suspension is there, and in those suspensions we have soil particles absorbed those actually absorb the hydrocarbons. So the soil particles is coated with hydrocarbons in Sundarban. Not only in Sundarban, in any natural soil system, if you add oil or any pollutant, these things happen. This is just a chemistry. And what we could show that the chemical oxygen demand of that water goes down once we actually inoculate that water, or rather we use that water as our medium for growing haloarchaeal species. So we looked into this, and then we thought, okay, so if it is the case, then we need to know why they are degrading hydrocarbons. So we moved on. We took the isolated organism, and then we moved towards analyzing the genome. And when we looked into the genome, we could see this is the pathway. Normally there are different types of hydrocarbons in nature. Now these hydrocarbons, when they are metabolized through a central molecule. Sometimes this is genticide, sometimes it is catechol. Again, this is chemistry, and it's an interface of chemistry and biology. So these central molecules are further cleaved by different types of dioxygenase enzymes present in different types of hydrocarbon degrading microorganisms. And then these downstream products that are being generated are, as you see, beta keto adipate and so on. They enter into the general metabolism that the organisms have. So when we looked into the genome, here are the examples of 3 genomes that we have performed. In one genome, you have only genticide degradation, genticide pathway, whereas in other genome, you have 2 different types of pathway to handle all the different types of hydrocarbons, confirming the fact that, well, in the genome, they are also equipped with machineries or enzymes that can allow them to degrade those hydrocarbons. Now, this actually led us to conclude that, well, in one hand, pollution is actually shifting the microbial communities in a given sediment, but such a shifting is actually allowing abundance of organisms that are capable of degrading those pollutants, thereby helping the environment, or helping the environment to come back to its original state. So the nature is working for itself, but we need to also assist, because these degradation processes are not very fast in nature. So they are trying, but if we stop adding oil, and if we process the oil or this type of hydrocarbons, or even sometimes heavy metals, uh, then there will be less shift of microbial community, because normal microbial communities are essential for maintenance of normal ecosystem. Let's say for mangrove, it's a mangrove ecosystem, there are processes going on, and for those processes you need a common microbial communities, which is being shifted due to the pollution. Although the shifted community is trying to restore the environment. Not only that, in a very recent study, we have found something extraordinary, that there are reports that heavy metals and hydrocarbon pollution in different types of environment around the world, they are helping in dissemination of antibiotic resistant genes. So when we found that we have lot of heavy metal and hydrocarbon pollution in Sundarban, we actually looked into that parameter too, whether the antibiotic resistant genes dissemination means the gene cassette from our antibiotic resistant gene cassette is shifting from a resistant organism to an organism which is non-resistant. It's a basically spreading of the antibiotic resistant genes across microbial communities, and that is being shown, or we have found that in regions where we have higher heavy metals and hydrocarbon pollution, we have higher concentration of one representative genes, which is basically BLATM, in this case ampicillin resistant genes. So our conclusions were, pollution not only alters microbial community diversity, but also helps in dissemination of antibiotic resistance in natural environment, which is very alarming, because till today, I think all of you know that in medical setup, clinical setup, we have antibiotic resistant genes dissemination is going on. Not only that, we see multi-drug resistant organisms in clinical setup. We report them, but very few studies from India at least available where people have shown that it is not only in a clinical setup but also in natural environment we are having this problem. So, and there are theories coming up. One of such theories is that co-evolution theory of heavy metal and antibiotic resistant genes in nature. So basically, heavy metals and antibiotic resistant antibiotics, they do use receptor molecules which are having similar epitopes. So thereby, so when you have heavy metal resistance in a given environment, that organism that resists to that heavy metal is actually also allowing the antibiotic resistance genes to come in and integrate into its genome through horizontal gene transfer, and thereby making that organism antibiotic resistant. So this is very alarming, and this is one of the very important aspect. Also, right now, in India, it's popularizing in research. Okay, so the take-home message is that when you look into the sediment, it is anaerobic sediment, so they're dominated by anaerobic organisms, and then you have aerobic rhizosphere, how I am showing you later. Then we have more diversity with lot of pollution. So more food, you provide more food, you have lot of diversity, and that lot of diversity also includes dominance of haloarchaea, at least in Sundarban. I'm talking about archaea part. Also in bacteria part, we do see a lot of changes going on. And to confirm that dominance of haloarchaea is related to this oil pollution or hydrocarbon pollution, we went for pure culture isolations. We looked into the reduction of chemical oxygen demand, and with that result, we can say we have a possible remediation agent, natural remediation agent that is probably operative in Sundarban-like environment. And there are multiple pathways of hydrocarbon degradations are identified, and we believe that this holistic view is actually giving us a clue that nature is trying to restore itself, but the input is much more than the organisms are capable to handle. So that is why the environment is getting degraded every time, every day. So we need more restoration effort to protect this wetland, mangrove wetland. So as I'm giving you the idea of how microbial communities are changing, let me talk about rhizosphere. So it's basically mangrove rhizosphere. You know about rhizosphere. It's a microecological zone. It's in direct proximity of plant root, and it is under influence of root exudates. So root exudates are playing very important role in shaping its own microbial diversity in the rhizosphere region, the rhizosphere microniche, and it is very unique soil system, different for overall surrounding bulk soil. So so in Sundarban, so we work at different places. See Kolos, Gotkalis, they are really distant. This one is inhabited place, this one is forested area, and we do work on two plants which are the most abundant ones. One is Gorad, Seriops decandra, and the vine which is Avicennia alba. These are the two plants where we looked into their rhizosphere microbial diversity. And interesting thing is when we looked into the rhizosphere microbiome, again, I don't want to go into the data. So let me explain what happened in bulk soil. It's basically anaerobic bulk soil, but what happens in from the root, we have release of oxygen, and that release of oxygen is actually making this region a pretty high with oxygen concentration. So it's more or less oxic rather than the bulk soil or the topsoil, which is anoxic because of regular inundation. So this rhizosphere region is more oxic, and that is directly reflected into the diversity of bacteria as well as archaea. So what we could see, unlike sediment, rhizosphere bacterial community is dominated by gamma proteobacteria, which indicates that we have anaerobic, not anaerobic environment, it is now aerobic environment. And we believe we have data I'm not showing you today. It's called rhizosphere effect, that rhizosphere root exudates are playing an essential role in shaping the microbial diversity or the community diversity in this area. And finally, irrespective of the pollution status, some mangrove plants maintain a steady resident microbiome. So, for example, when we talk about Goran plants, we could see that it is similar to sediment samples. We could see dominance of haloarchaeal species in the polluted region. Whereas when we looked into the vine plants, so these are the plants. When we looked into the vine plants, we could see it is controlling its own rhizosphere microbiome, irrespective of the pollution status. So this is really something we learned from our study, that indeed plants can also control the microbiota of given environment by releasing different components of root exudates and controlling the microbiome. Okay, so with a positive note, let me end my presentation is that microbes do adapt and you can use them. So this is one of the examples where we isolated a plant growth promoting microorganism from mangrove rhizosphere, and a number of actually organisms, and those let me tell you something, that Sundarban is known for its high concentration of heavy metals in the sediment. It's well known. Now the problem is there are very few studies in the content of these heavy metals in the crops made in the coastal area of West Bengal, for example. So we took an approach where we looked into the arsenic concentration in the crops, where we made a collaboration with Pollution Control Board, and we had these organisms which are plant growth promoting microorganisms, and they do actually assist plant growth. Not only that, they do absorb and accumulate arsenic on their membrane structure, and thereby allowing less amount of arsenic to go into the plant, and thereby the content in the of arsenic or arsenic salts, different types of insoluble salts of arsenic in the seed or the crop seed, that we could actually estimate it. Also this is an example of plant growth promoting organisms from hypersaline environment, because in coastal area, if you want to develop a biofertilizer, for example, you need to have an organism that function at elevated salt concentration, because in coastal area, due to inundations during the cyclones and so on, we have high salt concentration in the agricultural lands, and that's why this is very important to have some organisms with plant growth promoting attributes and abilities to survive at high elevated salt concentration. Okay, with this I would like to acknowledge this is my lab, my collaborators, and yeah, these are my
Thank you, sir.
Yes, please.
Thank you, Dr. Avrajyoti Ghosh, for your detailed discussion on the mangrove ecosystem. Now I request Dr. Abhishek Mukherjee to place the question to
Thank,
Dr. Avrajyoti Ghosh.
thank you, Bibasadil. The first question is, can the microbiota in a given environment get
Yeah.
totally altered after a natural calamity? For example, after the Ayla or Amphan, is there a possible of succession or the change in temporary and the original community comes back after the ecosystem is stabilized again?
Yes, it's a very good question actually. So when we had Ayla, at that time there were no reports on the microbial diversity of Sundarban. It was like some people isolated some organisms and they reported it. but the global or the holistic view by utilizing the modern bioinformatics and computational tool was not available at that time. So we had no clue how it was changed at that period of time. But yes, you can actually measure the changes, and people do it nowadays. It's a very common practice to measure the microbial. Actually, it's a routine monitoring of microbial community diversity in different environments before an event and after an event. This is very common, and so from those studies you can see the changes, and that will be directly, if you see also in Sundarban, from my experience for last 10 years, for example, uh, what I understood that in regions where you have more human intervention, you will see that the mangrove plants are dying, and very straight. If you go to, let's say, Gotkhali jetty area or Jharkhali jetty area, you will see near the jetty the condition of the plants are very bad because of regular spillage of oils and also human activities. So that is, I think that is also not only think, I have data to show that it's the microbes that change first because they are more susceptible. Once the microbiome is changing, that finally leads to the change in the plantation. Okay, so they are deeply associated. So once they are helping each other, so once you change or disturb the microbiota, it will change the plantation and also eventually change the entire stretch. Again, I have to mention that after Ayala, there were some places where they had restoration program. So government of West Bengal actually performed some restoration program in some areas, and there the microbial diversity is actually coming back to its original one. So the pristine area where from we analyze the microbiome, the same microbiome or same pattern of microbial community diversity you can see from the restoration area, which is a good in a sense that uh, you can actually see a restoration also in the microbial communities.
Okay. Okay, sir. The next question is, what may be the easiest way to cultivate any archaea in college laboratory just to show students show that they can grow their interest in that area?
Well, yeah. So you have to have a particular. So the main problem is the bottleneck is um, they are very abundant in nature, but the composition of their medium. I mean, the medium composition or the food they want to grow in the laboratory is quite unique, and there are available medium. For example, if you want to do haloarchaeal isolation, I can provide you some medium. There are some medium that people have standardized, but always you have to play around. Unlike bacteria, they are difficult to grow because they are slow growing, and as most of you know, they represent third domain of life. So evolutionary, they are very important, but unfortunately it's very difficult to grow them because of their selection of nutrients. Not all the nutrients they do utilize. For that, they have to, you have to actually play around with the medium, and you can isolate some. But there are some standard mediums that we also use, that I can share if you want to show students how you can isolate. But it is a time consuming one. For example, I'm 7 to 15 days you need to spend, and for practical classes I think this is quite a lot, so you have to plan accordingly. But yes, you can do it.
Oh, thank you, sir. The next question is from Devolina Bhattacharya. Is it possible to detect the heavy metal pollutants in the wastewater by forming bacterial enzymes like dioxygenase oxidoreductase with nanoparticle corona?
Well, that's a very difficult and tricky question. I don't know the answer. But yeah, so there are standard procedures for heavy metal pollution measurement. It's atomic absorption spectroscopy. You can have, I mean, this is very common instrument nowadays. So you can really do measure heavy metals in a routine basis. There are other instruments too. But regarding the question, Nanoparticle based estimation is still, I don't think they are in place. Maybe in research there are people working, but I am not aware of any technique, standardized technique for heavy metal pollution measurement with nanoparticles.
Okay. Thank you, sir. The next question is, can biofilm be isolated from the mangrove ecosystem? Is some work going on for biofilm isolation? How do we do that?
We will wait for some time. I think there is an network issue. We will wait till Dr. Ghosh returns. We will wait for some time. I think Dr. Ghosh can't be reached. Let's wait for some time. Mm,
I was disconnected.
yes, yes, sir. Yeah.
Okay.
So should I continue with the answer?
Yes, sir.
Yes,
The
yes, sir.
last question.
Yeah, so the point is, yes, biofilm, you can isolate the biofilm. Normally, if you isolate any biofilm from natural environment, it is a mixed biofilm. So normally researchers try to develop their own biofilm, because if you want to understand any particular feature of any given biofilm, then you need to know the content of that biofilm. And so the natural biofilms are not always, you have to first characterize them and you have to maintain them. Now that is kind of very difficult. So we do isolate organisms for different purposes, for different biotechnological purposes, and we do make biofilms. And we use the, actually there are two kinds of biofilm formation. One is static, one is dynamic, and we mostly go on with static biofilm development, and then we study different aspects that are related to the given environment. Yes, you can do it.
Okay, sir. Thank you. I think that's these are all the questions, and thank you for answering them. We will move. We are about to end our program. But before that, I would like to tell that there was also a poster competition was arranged today for the UG and PG students, and we had 46 participants, and already their posters are being analyzed, and the result will be declared on our college website today after 6 p.m. Okay. So next, I will request Dr. Shubhadeep Shamanto, Assistant Professor, Department of Chemistry, Maulana Azad College, to deliver the vote of thanks and to terminate the program.
Thank you very much, Vipasha. We are now in the closing session of today's National Science Day celebration, which is a DBT Star College program in collaboration with Microbiologist Society of India. It is a privilege to extend the formal vote of thanks of today's webinar on behalf of the entire organizing committee and the college. We had organized this webinar to commemorate one of the most significant discoveries of classical physics, the Raman effect, which perhaps changed forever the face of spectroscopy. At the onset, we must extend our heartfelt gratitude to Mr. Sudhir Sahu and Dr. Abhrojyoti Bose for their captivating talks in two very different but harmonized disciplines, carrying much of the essence of this year's Science Day celebration theme. We are really do hope to host you again in our college in the post-COVID real world. We also express our deepest gratitude to Dr. Shapna Mukherjee for her constant help and patronage to make this webinar a success. We are looking forward to organize more student-oriented activities in collaboration with Microbiology Society of India in future. We are especially grateful to our principal sir, Dr. Shubhashish Datto, for his unconditional support in all of our endeavors. Professor Shubhit Dasgupta has been our captain and mentor in all of our programs. We cannot think of organizing a single event without you, sir. Lastly, we would like to convey our special thanks to all the participants whose active and passionate presence had added life to the webinar. Participants are also requested to fill and submit the feedback forms already posted in the chat box. Again, a big thanks to all of you. Have a nice day ahead. Thank you.
Thank you. Thank you.
Thank you, sir. Thank you all.
Thank you,
Uh,
everybody.
Thank you.